Hello, my name is Evgeny Cherkashin, and I would like to present my talk entitled Construction Techniques of Baikal Microbiome Research Informational Computational Environment. I work in Mothur Institute of System Dynamics and Control Theory, Siberian branch of Russian Academy of Science. Microbiome is a set of microbes which are investigated in human body, animals, soils, or water. It is taken and then by chemical reactions converted to a set of genes which is synchronized and put into an environment which is almost always some kind of cloud system. Then it is processed by algorithms which are preliminary developed by scientists, and the results of processing form diagrams. And after that, the fundamental study goes to practical, trying to earn money from this, organize distant monitoring. There are two main ways of genome investigation. The first one is amplicon analysis. Amplicons, it's many times copied part of DNA or RNA, which then is synchronized and analyzed. The second one is shotgun analysis. In this case, all DNA is cracked by parts, and the main aim is to reconstruct the whole DNA. After reconstruction, the structure of genes, a number of more general analyses can be done. For example, taxonomic analysis, constructing phylogenetic trees, phenomics, where we try to understand the relations between various species of microbes, functional assignment for number of genes expressed in proteins. The aim of our research is to construct a part of this data processing environment devoted to Lake Baikal. To be more precise, is to construct a big data database, metadata storage, and adapters for expressing data conversion each stage of investigation. Visual construction of processing models, metadata inference unit, results and intermediate data visualization representation, data integration unit based on semantic web and linked open data to automatically create parts of investigations, a part of resulting papers. And at this time we consider amplicon data analysis according to MiSeq standard operation procedure. The main part is consists of these 8 stages. The first one is a reconstruction of coatings, breeding, removing primers, filtering, classifying unique sequences, alignment, the reference sequences to Silva database. After this alignment, we obtain taxons stored in database. Other filtering procedures, for example, chimera removal, classification of the resulting set of similar readings into operational taxonomic unit. We tried this procedure ourselves, spending about 1 month, and now we realize that we should refine our instruments. To do so, we decided to reconstruct the investigation that was made by our colleagues from Limnological Institute. All the samples were in the same time analyzed according to MiSeq standard operation procedure. In this slide you can see application of align sequence operation by Mothur software, where faster relevance are input parameters and this list of files are output parameters. The main problem is that each scientist should do this procedure manually or make scripts. After almost each stage of the execution, he must apply summary sequence with various parameters which are different for each previous operation. To evaluate set of readings, here we have a number of readings classified by different length of base pairs. For example, here we have 109 and here 176, and we should decide which classes we will keep and which will be dropped out. And this operation should be made after each filtering, each construction, some other operation. In the middle stage, the set of readings is converted to number of operational taxonomic units, and these are described by their taxons of various level of precision. Some can be recognized as species, some can be recognized. In these investigations, we determine bacteria which Illuminobacter. This bacteria is harmful for human beings and animals. First time it was determined in 2017 by one Japanese scientist. Here we have an example of phylogenetic analysis where each point on the circle corresponds to one operational taxonomic unit. It represents the relations between OTUs as an abstract taxonomic tree. The more general analysis operation is to classify places, having compared their microbiomes by similarity. To correctly interpret this diagram, we better relate with places where samples were taken. This set of samples have some kind of similarity. This diagram is called heat map diagrams. This diagram was created by R because R has much more power to apply different alignments. But in order to construct a diagram of good quality, a program should be written. At the first stage of our project, we suggested a tool for visual creation of operation sequences using data flow diagramming. We created translator from description from source code of Mothur. We taken declarative parts, converted into triples of the source description framework, and then by means of logical objects we created a source code of modules representing these blocks in RapidMiner Studio. In this year, we found that there is a Galaxy project which is very useful in representing investigations using popular notebook approach like Python Jupyter. But in order to use these tools, we should convert our sequences to Galaxy representation. There is already developed a tool set representing MiSeq standard operation procedures in Galaxy. It has XML description like this one, and we decided to use this description to refine our converter. We need to add additional modules which were not converted by authors to make the representation more complete. So we have two model data sources, one from Mothur's source code and the second one is a description. As of today, we have the following architecture of our infrastructure. It contains 9 modules. The central one is metadata and model storage, integrated with various ontology servers like DBpedia. Although there is a text indexing engine, authoring tool based on browser. It's also our branch of research and development, allowing user to build documents on the fly in the browser, integrating various data by means of linked open data technologies. Here we have data storage. In this year, the integration of Galaxy project is underway. It will be main component used by main user. The information interchange in this architecture is organized with usage resource description framework. It allows us globally identify resources and flexibly describe data. Another one advantage is usage of standard ontologies like doubling core, friend of a friend, provenance, DBpedia, Bibo, etc. In this slide we present transformation scenario as a logical object. This is an example of facade object. In this project we have a good result in analyzing different tools for model transformation, namely logtalk. It is flexible. It allows us to encapsulate knowledge behind predicates and objects. In next few months, we have to implement data flow transformations from RapidMiner Studio to Galaxy representation. We have to fit additional modules, which are summary and data visualization. This we will realize additional logical objects for translation plans, which account different patterns. The second problem which we will solve is to create more sophisticated source code parser, adjustment of our transforming tools to account all available information about Mothur model structure, which is partially done in November. And the third step is to upgrade Galaxy Notebook to serve linked open data. And also we need to write a handbook for logtalk language. In conclusion, I would like to say that we try to master biologist activities, investigate it, and find some regular patterns which will be accounted in our knowledge base transformation and metadata conversion. We have tested our transformation tools. We have prototype of interactive document authoring tools which will be adopted to the microbiome research domain. And our primary aim is to integrate with Galaxy project. You can get the whole presentation with this URL. And thank you for your interest in our project.